Any questions? Okay, we've had one lecture so far in lecture exam 2 material. That was, if you get your PowerPoint handout from, I think it's the week 4 PowerPoint, get it out. Because I don't got a lot of time and I gotta get you up to speed so we can continue on. There was one slide that had a graph that showed resting membrane potentials being polarized, and we call that minus 70. Does this look familiar on the whiteboard here? This is, again, you gotta have this known for the rest of lecture exam 2 material. Threshold is less negative than resting membrane potential. So any signals, like these green guys, that get it closer to this is excitatory. Any signals that make it further away from threshold is inhibitory. But if it hits threshold, it's all or nothing. You're going to have the all, and it'll go up and peak at about plus 30. These, again, are example numbers. Every neuron is different. Okay? Remember that action potentials are self-propagating. They do not fade over distance, and even if it's going over 3 feet, it still does not fade with distance. Graded potentials are localized. They do fade with distance. They can either be excitatory or inhibitory, depending on whether it's getting closer to threshold or not. Are we all together? Everybody see that graph?
Mm-hmm.
So if you go less negative, closer to threshold, that's depolarizing. Depolarizing is excitatory because you're closer to threshold. This concept, people, again, by lecture exam 2 here, I'm going to tell you something that you need to do and need to know. That concept has to become as natural to you as breathing by lecture exam 2. If you have to take time to review your notes to get it, this stuff is going to go up past your ears. That has to become as natural to you as breathing. If you get a signal that increases the negativity, it increases the polarity, that's hyperpolarized, you're further away from threshold, that's inhibitory. Everybody got that?
Mm-hmm.
Again, mark that next to that slide. As natural as breathing.
Breathing.
Okay. We just barely got on the topic of receptors, talked about transduction, specificity, receptor endings, and the law of specific nerve energies. And then I think we ended up here. Does this sound like about right?
Yeah.
Now, I'm putting, instead of number 4, I'm putting this under graded potentials, because these are actually graded potentials. You can see it, it's highlighted. It's a specific form of graded potentials that receptors do. Remember that transduction is taking an external energy, like light, with your rods and cones, and converting it to a neural event. The vast majority of transduction is this. Other receptors like tactile free nerve endings, it goes right to action potentials. So this is most transduction becomes receptor generator potential, and this is some transduction. These are actually the exceptions to the rule. The vast majority of transduction is this. And again, it's going to result in EPSP, which are graded potentials. That's why it's listed under there. But it's the EPSP that receptors do. We call those receptor or generated potentials. And because they're graded, they are localized. Over a little distance, it starts to fade. And the degree of depolarization is proportional to the stimulus intensity. Stronger the stimulus, the more depolarized it becomes. The quicker it gets to threshold. The stronger the stimulus, the more spikes will result. So we can see A, B, and C are different generator potentials down here. But the threshold, all receptors also have threshold. And if, say, C hits threshold, then you're gonna get a spike. We're gonna talk about tonic receptors and phasic receptors in just a little bit, which is also relevant for lab. In fact, a lot of today's lecture is relevant for lab 6 and 7 next week. A lot of what we're talking today is relevant for lab next week. And again, you see here what you have is stronger stimulus on the bottom. You're gonna get a more depolarized receptor potential. It's gonna result in more spikes. Everybody see that? Remember another thing I said last week that you might wanna write up here next to this top graph is our brains feel nothing. Our brains feel nothing. Sensory-wise, our brains only know one thing to do, and that is receive those green spikes. All right. Again, this threshold is an example. All neurons are different. And that is where we talk about recruitment. Talk about intensity coding as recruitment and frequency modulation. This top bullet is about recruitment. Different neurons have different thresholds. And the ones that are higher, say minus 50 millivolts, because that would be higher in the graph, will only respond to stronger stimuli. If it's just threshold to minus 55, it's not going to reach threshold. You actually, for those higher threshold neurons, it has to be more depolarized. The other form is frequency modulation. So this is a repeat of what we discussed in week 4. We mentioned recruitment and frequency modulation right here. Here they are again. Stronger the stimulus, that neuron is going to fire more spikes. Recruitment is more neuron firing spikes. Frequency modulation is about the one neuron firing more spikes. Recruitment is more neurons firing spikes.
Okay. Okay.
In fact, I would draw to the side of this slide these first 2 bullets and write intensity coding. That was from week 4. Intensity coding were these 2 bullets. And then we're going to do something in lab that has to do with what are called receptive fields. Next week's lab is actually probably the most fun lab. Because we get to actually experience some of the stuff I'm talking about. Keyword here, either write it down or circle it. A receptive field is an area supplied by a single sensory neuron. So here we have 2 sensory neurons. Here we have 1 sensory neuron. because it's a receptive field is supplied by a single sensory neuron. Down here we have two receptive fields, because it's a 1-to-1 ratio. Everybody following that? Well, we're gonna do a thing in lab next week called the 2-point discrimination test. If you go to areas that are real sensitive to touch, like on your fingertips, or on the outside of your lips, everybody, outside of the lips. They're really small receptive fields. You've got a lot of sensory neurons there. If you have the caliper points a certain distance away, like say here and here, because they're going up two different neurons, it's going to feel like 2 points. Then you go to the back, see the back of the base of your neck. Same distance. I'm going to ballpark it. But here, only one sensory neuron is taken into the brain, even though 2 points. The same distance as over here. This feels like 1 point.
Okay.
Why? Because your brains feel nothing. Your brain only knows to do one thing sensory-wise, and that is to receive spike. And if it's receiving spikes from one sensory neuron, it feels like 1 point, even though 2 points are touching. Because your brain feels nothing. Everybody got it? Are you following what I'm saying? That's all on the third one. Oh, that kind of looks like what I did on the whiteboard. Okay. Oh, that kind of looks like what I did on the whiteboard. Okay. All right, now we got to talk about sensory adaptation. Sensory adaptation is specifically this. With constant stimulus, you will get a decrease in action potential firing rate. That is sensory adaptation or sensory accommodation. Those are synonyms. You should write that down somewhere. Sensory adaptation or sensory accommodation. Same thing. So if a stimulus turns on here and turns off here, if you see action potentials, and that's each of these red spikes, really start off big, but then because the stimulus is constant, up here at the plateau, it slows down. This is sensory adaptation. Okay? Those that do it more are called phasic receptors.
More sensitive to changes than stimulus.
There are some, though, that don't do it very much, and they're called tonic receptors. Pretty much, if the stimulus is on, it fires. It might slow down a little bit. But it pretty much, if the stimulus is there, tonic receptors fire. Classic example of this are nociceptors, pain receptors. And if that toothache is hurting, it's going to hurt you all night. Okay?
What do you mean by the phasic receptors being more sensitive to changes in the stimulus than the stimulus itself?
Right?
I don't get it.
Like this. It fires here, while it's constant, it dies out. But hey, look what happens when it changes to off at full fire again. It's more sensitive to the vertical lines of the stimulus, boom and boom, than it is to this horizontal line.
Okay. Got it.
Can I give you a good example here? It's listed, thermoreceptors. Just think about a hot tub.
Yeah.
It's hot when you first get in, you stay in, pretty soon it doesn't feel so hot anymore. Until you get out, and then it feels cold.
Gotcha.
Everybody get that? Next to the first bullet, write hot tub. Draw a line, see where it says thermoreceptors. Convert as much as you can, people, and if you get into your programs, convert as much as you can of this content to the experience of being human. And you don't forget it. A lot of what I'm teaching is common sense, common to the human experience. First bullet, hot tub. Second bullet, that toothache that hurts all night. This is from your textbook showing you the same thing. The red line is my top graph. Okay. All right. So sensory adaptation is really seen in phasic receptors, not so much in tonic receptors. They will slope a little bit. This, again, people, reality is there's no black and white, it's all shades of gray. A lot of what I'm teaching you here is oversimplified to the point that you understand it, but it is oversimplified to the point of being a lie. There are no fully tonic receptors. They are just very little phasic. Does that make sense? Shades of gray, not black and white. All right, I'm gonna erase this. Does this look familiar? Just remember that these terms only apply to this synapse. If there's a synapse here, for that synapse, this red one's the presynaptic neuron. Everybody remember that? These terms are specific to this synapse. So let's talk about synapse. That second cell doesn't necessarily have to be a neuron. It can be a muscle cell or a gland. The presynaptic neuron, for this synapse, the relevant area is the axon terminal. For the postsynaptic neuron, it could either be a dendrite or a cell body. Remember, both dendrites and cell bodies are the input zones. Postsynaptic are the middle letters here. Postsynaptic, it's the effect on the red cell over there. Probably shouldn't have used red. What communicates from one cell to the next is neurotransmitters. And as this animation is showing, there are synaptic vesicles at the axon terminal. When the action potential reaches the axon terminal, there's an influx of calcium ion, and that triggers exocytosis. That releases the neurotransmitter without it ever having to cross the lipid bilayer. It has to diffuse across the synaptic cleft, which is the gap between the two cells. This is why on this drawing, the purple and the red do not touch. They get really close, they don't touch. They are not in physical contact. There is a gap called the synaptic cleft. Then it has to bind to neurotransmitter receivers on the postsynaptic cell. Everybody got this slide?
Mm-hmm.
What do those receptor sites look like? Here it is right here, receptor. Neurotransmitters fit right into the key. The lock is the neurotransmitter. And there's the lock right there, receptor sites. These are proteins that have the shape of the neurotransmitter on them. Now, depending on which ions move, it can be either green or red here. Everybody got that? Mm-hmm. They are not all excitatory, they are not all inhibitory. It's just what ions are moving and in what direction. In fact, there's another slide coming up, but let's make sure we write a little line off from this, either excitatory or inhibitory. Write a line up to the side and write, depends on which ions and which way they're moving. It depends on which ions, some are positive and some are negative, and which way they're moving. And then you just have to think mathematically. For example, let's look at this one on this picture. When acetylcholine binds to this acetylcholine receptor, it opens up sodium channels. Since there's more sodium in the ECF, that should have been in your bonus essay from last week, it's facilitated diffusion. It's got to go from high to low. Since there's more sodium on the outside, it's got to go from the outside ECF into the inside of that neuron. If you are adding positive ions, is that going to be green or red? You got to think mathematically.
Okay, adding positive.
Green.
It's going to be green. If you add positive ions, you're going to make this less negative. Less negative is getting you closer to threshold. That's why that's excitatory, which is what the E means over there in green. As natural as breathing.
Mm-hmm.
You get what I'm saying now?
Mm-hmm.
As natural as breathing. Now, the time that the neurotransmitter is having an effect on the postsynaptic cell or neuron is extremely brief. That's what it's saying here. There's a couple different ways that your body gets rid of neurotransmitter in the synaptic cleft. This is only one of them, deactivating enzymes. Some presynaptic neurons take back in, by endocytosis, the neurotransmitter just spit out by exocytosis. Others just break it down and make more. This second bullet is about those neurons that just break it down to make more neurotransmitters. In anatomy, when you did the sliding filament theory, and to shut the action potential off, you need acetylcholinesterase. Does this ring a bell at all, anatomy students? Acetylcholinesterase belongs in the second bullet. It's down here at the bottom. That is an enzyme that breaks down acetylcholine into the two carbon units, the acetyl group, and choline.
Okay.
That's what it's showing you in this animation. Acetylcholinesterase breaks it into acetyl group and choline,
Oh, I see it splitting.
and then it falls off the receptor site. Everybody see that? All right, well, this whole thing about getting an influx of calcium to trigger exocytosis, to diffuse the synaptic cleft, and to generate threshold to trigger on another action potential takes time. Look at this animation. It's excellent for showing this. When you get to a synapse, it always slows things down. This is actually relevant for this week's lab. The more synapses you have, the slower the transmission. Look at how this, as soon as it gets to the synapse, it slows down. Boom, boom. Delay and delay. Delay and delay. You see how whenever you get to a synapse, it slows it down. It's called synaptic delay. And it's because it takes time for all these things. Exocytosis, diffusion, reaching threshold in the postsynaptic neuron. This topic is relevant from everything from aging to neurological diseases to various pharmacological effects. Even to the ability to hit a fast pitch baseball. Let's do reaction times, which is one thing we're doing in lab this week. Just as an interesting aside, that's why it shows a picture of Roger Clemens. Who threw a fast pitch baseball and cheated with performance enhancing drugs. This bottom sentence really applies to Roger Clemens. All right, now we've already actually covered this. We're just stating it in different ways for different perspectives. This synapse could either be excitatory, resulting in a depolarizing EPSP, or inhibitory, with a hyperpolarizing IPSP. And again, look at this figure. Inside the neuron is below the membrane in both situations. Everybody see that? Like I would write that. I would write out here, in here. Out here, in here. If you are adding that positive sodium, that's depolarizing, getting less negative because you're adding positives. Less negative is depolarizing, you're closer to threshold, it's excitatory. Now look at the bottom one. You're adding in negative chloride. If you add in a negative ion, it's getting more negative. You're further away from threshold, it's gonna get to -70. That's why the IPSP is inhibitory. You're now further away from threshold. Okay? This is going to be very relevant in the CNS. Everybody got this? Synaptic integration is what's happening on the postsynaptic neuron. Synaptic integration is about the red neuron over there. Because even though I drew one synapse, everybody see that? In reality, in your CNS, multipolar neurons can have tens of thousands of synapses on them between all the dendrites and all over the cell body. Look at this picture on this slide. If this was in reality, you would see no yellow. You would be completely covered with axon terminals to the point where you could not even see the neuron. That's reality. This is just oversimplified lie so that you're going to understand this. Does everybody get that? But they're painting in both green and red. That's all you would see is green and red. You would not see yellow. It is covered. So again, we're talking about the synapse from two different points of view. From the presynaptic perspective is what is the neurotransmitter. From the postsynaptic perspective is does this result in ions moving so that you depolarize, EPSP, or hyperpolarize, IPSP. And the key point for the postsynaptic neuron is how all these synapses all come together at the axon hillock. Do you hit threshold or not at the axon hillock? Then we are concentrating on multipolar neuron physiology. It is different for unipolar and bipolar neurons. Again, if this was reality, all you would see would be green and red, you would see no yellow. It would be completely covered. All right. So what happens to this red neuron when it's getting all these synapses? It is going to summate all these synapses because it's covered. What is the red neuron going to do with all these synapses? It is going to summate all these synapses at the axon hillock. This is where summation is relevant. And the question of summation is, do you hit threshold or not? Okay? There are two types of summation. There is spatial and temporal. I think we've covered that already. You guys remember that?
I don't.
All right, see all the purple presynaptic axon terminals I added? They're all in different spaces on the red one.
Okay.
That would be spatial summation.
Okay.
If this one synapse right here that I drew completely is excitatory, this guy can fire again and again and again and again over time. And you could summate and reach threshold in temporal summation, because temporal means time.
Okay.
Does that ring a bell? You better go back, especially if you did not do well on lecture exam 1 and you are in a grade hole. You better study every week like I'm testing you on Monday. Now this is fortunate in that this is only the second lecture of lecture exam 2, so you don't have that much to go back on. Go back on it like I'm testing you on Monday. Who does that? People who are serious about pursuing their careers in healthcare who are not academically brilliant. Who doesn't have to do that? Those that are academically brilliant. Look at your score in lecture exam 1 and evaluate for yourself if you are academically brilliant or not. And if you are not, I'm telling you what you need to do to succeed. And it's not difficult, it's just a pain in the ass hard work. If you are not academically brilliant and you are not willing to work at it, do something else. And I'm doing that because I love you. I'm saying that to you because I love you. Teachers who do not say this to you don't give a shit about you. And you're going to spend years and thousands of dollars wasted just to flunk out of pharmacology and you're out of the program. Find out if you're academically brilliant or not. And if you are not, be glad. Because now you can learn the discipline that'll make you a better healthcare provider. And again, it's all or none over there at the Axon Hillock, threshold or not. Okay, let's talk about various drug effects at the synapse. And this is actually how we began pharmacology, because we actually saw actual physical effects from various chemicals. And all the chemicals that originally became drugs were all natural. We didn't start making synthetic drugs until second half of the 20th century. For centuries, the whole understanding of pharmacology came from naturally occurring chemicals. Like crotoxin. Crotoxin comes from American rattlesnake venom. Botulinum toxin comes from botulism, black plague. They both inhibit the release of acetylcholine. That's why we call them inhibitors. It inhibits the release of acetylcholine, which is the neurotransmitter at neuromuscular junctions. So basically, this shuts off your skeletal muscle. Okay? The way rattlesnake poison kills you is you're not able to breathe because you can't do this. You have to contract skeletal muscle to inhale. And if you have too much crotoxin, you can't do this. You just get paralyzed and you die of suffocation because you can't inhale. Everybody got it? Same thing of the Black Plague. It would kill you because you can't do this. Interesting thing is we've taken that and we turned it into a product for plastic surgery reasons.
Fabulous.
You inject it in certain places in your face and you paralyze the facial muscles that when it contracts causes wrinkles. It's a microdose and it's very localized. When these things kill you, it's because it's gone systemic. Does everybody here know what those terms mean?
Yeah.
Eventually you do. Localized is just one little area, like right here where the crow's feet are. Systemic means it's going whole body. When an infection is localized, it starts swelling and it gets red right there, wherever the infection is. What's a sign when an infection is systemic?
Fever.
You get a fever. Okay. All right. A lot of aging actors have used Botox. But if you think about their profession, that's kind of, at a certain point, it becomes a point of diminishing returns.
Yeah.
Because if you paralyze your face, when you're really happy, you look like this. When you're really mad, you look like this. When you're really upset, you look like this. And when you're really sad, you look like this. certain point, you gotta shut off the Botox, or you can't make facial expressions happen.
I'm all for it.
All right. Again, neurotransmitters are all about causing ion channels to open. Well, if you block the ion channels, that chemical is a blocking agent. So even though, yeah, the neurotransmitter is released, ions can't move because the channels are blocked. Saxitoxin from red tide algae and tetrodotoxin from the pufferfish are examples of this. Japanese actually like to eat pufferfish just before it goes toxic.
Oh my gosh.
It tastes great, really great. It's the best sashimi and sushi ever. It doesn't really taste that good, you know, off season, but as it gets closer to the point where it's gonna kill you, it tastes better and better and better. But then if you eat it too late, it'll kill you.
Oh, that's a horrible risk.
How do you know?
Japanese word.
How do you know when it's too late?
Huh?
You're dead?
How do you know when like it's before it gets too late? It turns
Um,
colors or something?
No. You just, the people who are preparing it just have to be expert at it.
What's the red tide algae?
Yeah, there's a certain time of year in certain regions of the world where you cannot eat any of the fish that comes out of those waters. Because they have red tide algae, they have the saxitoxin in them.
Huh.
And again, if you block sodium channels, it's like you're paralyzed. It'll have the same effect as an inhibitor, because the ions can't move. Then you have enzyme destroyers, like sarin nerve gas. 25 years ago, there was an episode in the Tokyo subway where this cult released sarin nerve gas, and 12 people died, and several hundreds got close to it but recovered. Everybody got this?
So this is about like actually getting rid of the acetylcholine rather than.
Yes.
Okay.
And then let me just give you just a little bit of connection between physiology and pharmacology. You guys ready?
Yes.
The connection is sequential thinking,
Shoot.
which is why sequential thinking helps. I use that for the 5-point Canvas assignment. By the way, a student came in today, and this applies. If you sent the email but do not have the 5 points extra credit, I did not receive the email. All you have to do is bring up your device, show me the email you sent. It'll be in your sent box. If it was sent before the deadline, I'll give you the 5 points even though it's after the deadline. That's the best thing about digital media. It's time-stamped. Everybody got it? It's not a big deal. And you can't go back in time and generate the timestamp. So if you fell asleep at the wheel, it doesn't matter. But here's the thing. There are 2 ways to suffocate. Everybody look at me, because this applies to this section here. One is the inability to contract skeletal muscle. If you can't do this. Everybody got it? If you can't inhale, you suffocate. That makes sense, right?
Yeah.
The other thing is you can suffocate if you can't shut off the contraction. Because to not suffocate, you have to both inhale and exhale. Well, when you got this, working on acetylcholine especially, you're gonna do this, and then you can't shut it off. And you cannot inhale again until you exhale. You stay fully inflated and still suffocate, because you can't exhale unless you can shut off the muscles. Does that make sense? That's sequential. The sequence of the physiology of breathing. So that's what it is here. Keeps acetylcholine in the synaptic cleft. Keeps skeletal muscle in spastic contraction. You can't exhale. You still die of suffocation. Is this helping at all, or shall we just fill in the blanks?
No, it's
Because I'm still
great.
recovering. I could just rather just you guys fill in the blanks. I'm freaking exhausted.
Keep going.
And I'm too tired to beg you to pass this class.
So when you say other neurotoxins, is everything that you've listed so far technically a neurotoxin?
Yeah,
Okay.
so far these have been all neurotoxins.
Rat poison. Oh my god.
Pest control companies don't use strychnine anymore. It's pretty rare. Too many cats and dogs ate the rat poison. So they don't use that anymore. There's other things they can use. Same thing though with strychnine, it interferes with turning things off. Spastic paralysis and asphyxiation. Though the interesting thing up here in the top bullet is that neostigmine actually does the same thing, but you can control the dose, and for someone with this problem, myasthenia gravis, they have trouble contracting muscles. If you give them controlled doses of neostigmine and inhibit acetylcholinesterase, they can have more muscle contraction that's already deficient. So we can use these things that can cause problems and actually treat people that have different problems. Does this make sense?
Mm-hmm.
Oh, this up here? See this figure?
Yeah.
That's pharmacology. So you think this class is hard? Do you see why I'm as difficult as I am?
Mm-hmm.
It's because I know what's coming up against you. My job is to get you prepared so that no person in any program is going, the hell kind of A&P teacher did you have? Kind of like I was saying, you guys need to write your high school strongly worded letter. I never want to contribute to that. I'm going to get you ready, whether you like it or not. And if you don't want to do it, then you will not pass this class. Okay. Cocaine. Let's talk about cocaine.
Okay.
All right, let's convert all this information on cocaine to the experience all that hopefully not many of you have had. But I went to UCLA in the 80s, man. I mean, that was the peak of cocaine. And your classic example that you will experience is that someone at the party is like the life of the party. A lot of fun, a lot of energy, just having a great time. In 3 hours, he's a raging asshole. That's your experience if you've ever been in a party with someone on cocaine. Well, now let's look at the pharmacology. Cocaine, the neurotransmitter here are dopamine neurotransmitters, which are used in the pleasure centers of the brain. This is one example here where it's not an enzyme deactivation, but the purple neurons releasing dopamine take it back right away. Just releases dopamine, takes it back. Releases dopamine, takes it back. Everybody got it? In the pleasure centers. Well, when you block the reuptake channels, the dopamine stays out in the synaptic clefts, in the pleasure centers, which is why this person is feeling great, is the life of the party, a lot of fun. Everybody got it?
Mm-hmm.
Well, the thing is that cocaine is actually made from an amino acid, tyrosine, and eventually you're gonna deplete the tyrosine and the dopamine. The moods in the brain are a complex balance of different neurotransmitters. So when someone is bipolar, so someone has clinical pharmacological depression, they're gonna take medications that are adjusting the neurotransmitter ratios in the brain. Everybody got it? Well, you don't have enough dopamine because it's just depleted since all these neurons weren't able to take it back up, and you become a raging asshole.
Dang.
Okay, that is pharmacology taught really straightforward in a way that is something that maybe you have seen. Okay? Any questions? That's all that's in this bullet. All right, there's a ton of neurotransmitters, there's over 30. I'm going to hold you to what I cover in the PowerPoint. Set it up like an outline.
Oh boy.
Acetylcholine is always excitatory in somatic motor, because the skeletal muscle is the only somatic motor you have. But in autonomic, it could be excitatory or inhibitory. Acetylcholine is used by all preganglionic autonomic neurons, but only the postganglionic parasympathetic neurons. How many slides do I have to get to?
Oh boy.
Yeah, I'm gonna cover this in more detail later. I know this probably doesn't mean too much to you right now, but we'll come back to this when I actually get to this point in the autonomic nervous system. Okay? But just know in somatic, those are the things you're conscious of. In somatic motor, it's only excitatory. In autonomic motor, it could be either. And this is a table from your textbook. Then you have monoamines. That means they come from one amino acid. Sometimes they'll pair up two different ones, but they're really small. It's derived from amino acids, monoamines. What I wrote in this bullet is that these are broken down by deactivating enzymes. And the deactivating enzyme is called monoamine oxidase. Which, by the way, is one of the things that psychiatrists use to help find neurotransmitter balance in people. People that either have bipolar conditions or depression. Some of the drugs are monoamine oxidase inhibitors. Okay. You have serotonin. Immunosity comes from the tryptophan. This is one of the neurotransmitters in the mix with dopamine for finding a good mood balance. When you have a serotonin deficiency, sometimes they will give you a drug like Zoloft, which is a serotonin monoamine oxidase inhibitor. So if you have a deficiency of serotonin, and you're gonna now interfere with breaking it down, the little bit that you got will have a better, bigger effect. Now it's not so deficient. Do I need to repeat that? Zoloft, everybody knows, has been used for depression. Okay, not everybody, but if you're going into healthcare, you should. It is a monoamine oxidase inhibitor. What it is from various tests, they are able to determine that you have a serotonin deficiency. they can't get you more serotonin. All they can get you is more tryptophan. Well, if you don't have a deficiency in tryptophan, giving you more is not gonna do you any good. If your brain is only gonna make so much serotonin, take some Zoloft. Now, when it's released, it stays in the synaptic cleft longer because this enzyme is being inhibited from breaking it down. You're getting a bigger effect from the little serotonin your brain makes, and you're able to now find balance. Does that make sense now?
Yes.
Does this talk make any of you want to become physicians? Here's the thing, and I know this in every class that I have, there are students that are smart enough to be giving the doctor's order, not just filling them. And then I am in the same classroom with people that are not in the ballpark and can't see the ballpark. I see the whole spectrum and I recognize the whole spectrum. All right, remember a long time ago we talked about cyclic AMP?
Yes.
Cyclic AMP is relevant here. These neurotransmitters actually don't enter the cell, they bind to receptors, and they cause the cells to change by cyclic AMP. Catecholamines, they are not serotonin, they are different. Serotonin comes from tryptophan, these come from tyrosine. There's a whole bunch of them. There's the ones you have to know. But serotonin is a monoamine because it comes from amino acid tryptophan. Catecholamines come from tyrosine. Different amino acid. You can see the sequence. It's an enzyme that turns tyrosine into L-DOPA, another enzyme turns it into dopamine, another enzyme turns it into norepinephrine, another enzyme turns norepinephrine to epinephrine. So epinephrine, or adrenaline, is actually the final product. Okay? Any questions? By the way, epinephrine and norepinephrine both are chemicals, the identical chemicals, both a hormone and a neurotransmitter. It just depends on how and when it's released. It could be either. And you should get used to the idea that the nervous system and the endocrine system is the same system. Even though they're in different chapters in your textbook, it's the same system. The specialty in medicine is called neuroendocrinology because it's the same system. And the same chemicals can be either neurotransmitters or hormones. Go ahead and fill in the blanks here.
There's a lot of fun stuff. Fun good stuff in this. I'm lost. Exciting.
This is help when I'm filling up the whiteboard.
Yes. Absolutely. Bless
Still
you.
waiting for someone to say no, and then I can stop doing it.
No way.
For lecture exam 2, you need to know this slide backwards and forwards. Not all of it, I'll explain in a second, but this is a potential short essay. Here is where you're going to put together which ions and which direction are they going.
Okay.
Write that down. Which ions and which directions are they going? First of all, this is all in the central nervous system. If it's excitatory, is it hyperpolarizing or depolarizing? If it's excitatory, depolarizing. It's got to get less negative. So, if these are positive ions, and also these ions are higher in the ECF, when you open up those channels, they go in. You better write that at the bottom of the first bullet. Those positive ions go in to the neuron. Since they are positive, that's why they are depolarizing excitatory. They're getting closer to threshold. Now for the bottom bullet, you've got to get more negative. Inhibitory is hyperpolarized. Well, here now you have to pay attention. The negative ion chloride is going in. It's making it more negative. And this is how I said you have to think mathematically on this. But there is more potassium in the S bonus question for your lecture exam 1. There's more potassium inside the cell. There's more potassium in the neuron. When they open, that potassium is going to leave. Follow me?
Mm-hmm.
If you lose positive ion, you get more negative.
Right.
If you lose positive ion, you get more negative. That's why it says it's going to cause chloride, either chloride to enter or potassium to leave. Both are going to make the neuron more negative, hyperpolarizing, inhibitory.
Makes sense.
Everybody got that?
Uh-huh.
Any question about that? This is a classic example of how this class sets up more complicated classes later. It is simple, even though, yeah, I'm lying to you. I'm making it simple.
Mm-hmm.
You just have to drill on the simple so that you know it. Just showing you chloride channel. That's causing chloride to enter. So look at the voltmeter, you can see it's getting more negative, it's going down from chloride going in. Or potassium can go out, it still goes down. Everybody see that in these two slides? You're adding a negative ion, more negative. You're losing positive ion, more negative.
Yep.
Everybody see that? Not complicated. All right, we're getting ready to go into a discussion on the brain, which I'll set up at the beginning of lecture on Wednesday. For right now, you've got to know about the blood-brain barrier and CSF, and that's all we're going to cover for the rest of today. There's a lot to the blood-brain barrier. What you need to know for these classes is two things. The endothelial cells of brain capillaries are zip tight to one another with tight junctions, which are like Ziploc. Zip. Then the capillary is covered with astrocytes, and any chemicals that go in and out has to get through both. This is basically a good picture here. Once all the foot plates of the astrocytes are on the capillary, right here, look at those pictures on the bottom.
Mm-hmm.
Once all the foot plates of all the astrocytes are on this capillary, you see no yellow. All you see is gray. And stuff has to get past it. Any questions? That is also relevant for pharmacology, for any drugs you want to get in the brain. I ain't done yet. Again, if this was accurate, you'd see only green, you wouldn't see the yellow. What's on the other side of the blood-brain barrier is CSF. fill in these blanks, and now we are done. I'll explain this at the beginning of Wednesday's lecture.